done in tree hoppers in my advisor's husband's lab right next door to me that looked at how experience affected tree hopper preferences. And there's also been studies in species like birds, like this female pied flycatcher. And it's important to remember at this point that preferences aren't only shown in animals, they've actually found that there are female preference effects in fungi and plants as well as animals. So, environmental variation in nighttime light levels, so our normal phases of the moon, affect behavior in many nocturnal animals. This includes the vocalization patterns, movement, female preferences, a lot of different behaviors that this can be found to affect in a bunch of different species. So if mate choice behavior differs under light conditions, then the strength and direction of sexual selection may vary, which may help explain why there is this maintenance of variation. So my first chapter looked at moon phase and reproductive behavior, so just reproductive behavior in both males and females across the natural nocturnal phases of the moon. My second chapter jumps into female preferences and choosiness across the natural nocturnal light levels of the moon phases. And then the third looks at that push in light caused by urbanization by us as humans and anthropogenic light pollution and how that affects their preferences and choosiness. And we did this in our very cute study species, which is the eastern gray tree frog. I love versicolor. So, variation in light levels could affect female reproductive behavior in three distinct ways. The first is that females may only approach breeding sites under certain phases of the moon or certain light conditions. The second is that female preferences may change under these different light conditions, and we define female preferences or preference functions as what the female likes. And her choosiness could also vary under these different phases and light conditions. And we define choosiness as how much the female likes it. I'll dive into that a little bit further in the presentation. But for now, we're going to focus for the first chapter on our first effect, which is that females may only be approaching the breeding sites under these certain light conditions. So, my first experiment asked the question, does moon phase affect reproductive behavior? And our hypothesis was that there are lunar effects on breeding occurrence and intensity. So whether they are showing up to breed and how heavily are they showing up to breed, how many of these species are coming out. So we had two main hypotheses. The first was the predator avoidance hypothesis, was that early predator detection is predicted to be the most effective during bright nights and the least effective during dim lights. So essentially, if you are a prey item, you are able to see your predator better if it is brighter. You can see them across the pond, you're able to visualize them, and because of that, you're able to avoid them. The second hypothesis was the reproductive synchronization hypothesis, which is the idea that reproductive synchronization may increase success of reproduction by maximizing the number of breeding adults in an aggregation while minimizing the predation effect via the dilution effect, which basically says that if you are in a group and there's more people, more individuals in your group, it's going to decrease your chance of being eaten by a predator. You're nice and snug in your little group, you're herd, and you are safer from that predator getting you. And it's important to note at this point that this prediction is rarely tested because it requires you as a scientist to try to study both males and females in the same night in a field season night, which is very difficult when you are out in the middle of the pond in the middle of the night and you're trying to look at both males and females. It gets pretty complicated. So I had some predictions based on these hypotheses for both breeding occurrence and intensity. The first for our predator avoidance hypothesis is that variation in nighttime light levels should drive variation in behavior. So we should see different kinds of behavior across these different phases of the moon. For our reproductive synchronization hypothesis, we predict that frogs should show increased reproductive activity during certain times in the lunar cycle. And because it is synchronization and reproduction, males and females should be responding in very similar ways to the same lunar cues. So basically, we want males and females to show the same type of behavior across these different light conditions. So all of this research was conducted at Byers Pond, which looks a bit like this. I promise you it's a lot scarier at night. And this pond is located right next to our field station up in Saukville, Wisconsin, which is about 45 pond is in the middle of the woods, and when you're going in the middle of the night, it gets pretty hairy out there. So, when we go out to the pond, I like to put this picture up and play a little find the frog game. It is night, and our frogs blend in very well to their environment, so it can be kind of hard to see them when it is very dark. And you can see this one is hiding right there. And our main way that we end up finding them in the water is using a headlamp on the top, which I'm using to kind of show our frog right there. And we actually look at the surface of the water and look for their eye spots and the reflection of their eyes back at us. It takes a lot of practice, and I got pretty good at it at the end, but it definitely is a learning curve to try to find these guys at the very beginning. But sometimes they do make it kind of easy for us and live up to their name. As you can see, our little eastern gray tree frog is living up to their name as a tree frog in this picture. There we go. So at this point, I made this handy dandy video in my last field season, and I like to show you guys kind of what it sounds and feels like when you're at the pond. So this would be a very high chorus note. This is a lot of males at the beginning of our season coming out and singing their little hearts out to all their females. And it is deafening. And it's about this level too. So that I would characterize as a high chorus night. That is a number 4 on a 1 to 4 scale. That is our males at the very beginning being very motivated to try to get a female. And it is deafening, and it is very dark out there. So my first chapter asks the question, does moon phase affect reproductive behavior? Well, we sampled male chorusing behavior over 3 seasons, from May until July, which is our typical field season, from the years of 2011 to 2013. And this chorusing data was taken from a long-term recorder that's set out at our field site from a little bit before our field season to about a month after. And I sampled that over all of the recordings that we had of that year, from 2011 to 2013. And then we have our female breeding activity, which was sampled over 7 seasons, from May to July, again, our normal field season, from 2009 to 2015. Now, it is important at this point to look at the difference in the number of seasons. We didn't always have that long-term recorder out there. That was something my advisor started a couple years later and was not really extended for that long. So I had a little bit less data to work with on our males than our females. And our female breeding activity was figured out by the number of females that were caught in a collection night, which was always recorded across our field seasons. Now, this particular part of my research was actually done in the first year of my master's before I had ever stepped in a pond. So, unfortunately, I was not able to have illumination and moon phase data from stuff actually collected from the field. We have different light sensors that can be taken out there to see the actual illumination. So this was all taken from a website, staging time and date, that we figured out our illumination levels in our moon phase and paired that with our dates of collection. So, got a little ahead of myself. So starting out, we looked and separated it between males and females. So starting first with our males, does moon phase affect male reproductive behavior? Well, after our study, I compiled all this data into this very, very complicated graph. So I'll take and break that down for you guys for a second. Our occurrence of chorusing, so whether the males showed up on a night, is in gray and can be represented by our gray line. And our intensity of the chorus, how many were there? Remember that high intensity chorus night that I showed you guys? Is in black. So we found that across the natural nocturnal light levels from 0 to 100, that there is a likelihood of chorus forming, and the size of the chorus was found to be larger at intermediately bright light levels. So you can see that we have a dip down, a dip down on our less bright nights, and a dip down on our brighter nights. However, we see that peak in our moderate light levels. We also did another form of graphing where we put this on a 360-degree axis, and I have plotted the phases of the moon to make it a little bit easier to understand. And then we put a vector in to see where it was more likely to occur, during that first quarter of the moon. So you can see that little tiny arrow directing towards our first quarter of the moon. In addition to that, our larger choruses were tending to form during the first quarter of the moon as well. So you can see our males also showed a more intense chorus during that phase of the moon. So moving on to our females, whether females came to breed or not was not really affected by light levels. So you can see again in our incurrence of breeding, we have a pretty low peak in the moderately bright nights. However, it stays relatively flat as far as our females showing up to breed. However, there was a small peak in our intensity of breeding during the moderately bright nights. Female breeding activity was somewhat more likely to occur during the first quarter, but you can see if you look at this compared to our males, we're pretty uniformly distributed across the different phases of the moon. And you can see our arrow is a lot smaller for that effect. So they're maybe showing up a little bit more in the first phase of the moon, however, it's not super significant. And we found that our breeding intensity peaked around the third quarter. You can see we had one really intense collection night during the third quarter that kind of shifted our data. However, again, that very weak line towards the third quarter of the moon. So not as an intense of an effect in our females as it was in our males. So from this, I concluded that reproductive activity was somewhat weakly affected by lunar cues, with the effect being somewhat more strong in males than females. So going back to our hypotheses, our predator avoidance hypotheses for our males, we actually did not find support for, because remember, our males were the most active during those moderately bright nights. Now, in this case, we would want our males to be the most effective and the most intense breeding occurring during those bright nights, so when they're able to see their predators. So that doesn't really fit our predator avoidance hypothesis. However, our females may show some kind of trade-off, because our females move about the pond pretty stealthily, so they kind of go through the water to find their males. And they are neither active in the bright nights and not very active during our less bright nights. So maybe that moderately bright night may be a trade-off for her to get her breeding in and still stay somewhat conspicuous to predators. So maybe some weak effects for females for that hypothesis. As far as the reproductive synchronization hypothesis goes, we flat out reject that. You saw in our first set of graphs that we had completely different periods of the moon phases, but our males were active as compared to our females. And remember, we wanted that synchronization of their behavior across the different phases. So a little bit of effect for females for our predator avoidance, but not a ton of support. And I have made all three of these papers available for the department, so if you guys have any more that you want to read about this, you can find that in my first paper. So, going back to our three effects on female reproductive behavior, well, we've already found that there's not really any proof that females are approaching the breeding site under certain light conditions, so we can kind of forget about that and move on to female preferences changing under different light conditions and choosiness varying under these different light conditions as well. So, before I dive into preferences, I want to give you guys more of an understanding of how female preference functions actually are working. So in this experiment, we have a single speaker trial. So we put the female in a chamber, which I'll show you guys in a little bit what that looks like. And we ask her to just make one choice. We give her one call across the speaker, and we see how she responds to 9 different call duration stimuli, anywhere from 6 pulses up to 30. So a 6-pulse call is going to be an unattractive male, and a 30-pulse call is going to be a very attractive male. And what we see is that the female response is going to increase across that male trait. So as the male trait gets more desirable, that longer call, she's going to have a higher response to that. So we show her, sorry, that was a little loud. We show her an unattractive male, and we show her a really attractive male. I actually had to go next door to my teenage neighbor and ask her who an attractive male and an unattractive male celebrity was, because I used to have Ryan Reynolds and Zach Galifianakis to our preference functions, we also look at her specific traits of her preference. So we look at her peak, her highest response that she has across that male trait, her strength, so how is she able to differ between those high and low quality traits, her tolerance, so the response that she gets a relatively high, the trait that she gets a relatively high response to, and then her overall level of responsiveness across her preference function. Now, choosiness is a little bit different. We give her a choice in this case. So we have two speakers that we play her two different alternating calls from, and we present her with an attractive call that is attenuated, and then an unattractive call that stays. So an attenuation is when you lower the decibels of that call, so you make that male seem further and further away from our female, while we keep the attractive call at the same distance, and see at which point she goes, he's not worth making the effort for. So again, going back to our unattractive and attractive males, we ask our female to choose between them, and when they're both the same distance apart, I mean, come on, of course you're gonna go towards that attractive male. However, we look to see, pulling him a little bit further away, so your boyfriend is, instead of being at Hope, is across the state. He's not right there with you. But I guess you'll still stay with him, right? But he's in a different state. He's too far away, and you've got this guy that keeps bothering you, and I guess at some point you go, okay. I guess. So we ask with an unattractive and attractive male, well, what is she gonna do? And then finally, to be all-encompassing in what we look at her behavior, we like to look at some other finer scale approach behaviors. So females, when they are choosing, have some different movements such as hopping versus crawling. And then we also look at her latency to choose, how long it takes her to get to her choice, as well as her latency to leave the release box that we have her in and make that first move towards her choice. So this is where I like to show you guys kind of what our trials look like, and for whatever reason there's this goofy green in the beginning of this video. It goes away in a second, but I could not seem to get rid of it in here. But we have our trial start out, and this is our chamber that we do our testing in. We put her in a release box right here, and then we start the time when we pull up our pulley, and there is a speaker right here that we are playing her a call. These recordings do not have audio with them, so she's hearing a call right now, and you can see she's moving towards her choice, and we mark her choice as landing in this choice zone in front of our speaker. And you can see as she's moving, she's making these small adjustments, she's rotating her head, and then she has made her choice in that call. So after field season is over, after I'm done smelling like pond scum for 2 months, at the very end of the season, we compile all of our videos into an image so we can see her path, as well as keeping those videos so we can look at those finer scale approach behaviors, the movement of her head, her hopping, her crawling, and we do a lot of analysis. Let me tell you, stacking all of those videos into this one image takes like 5 minutes a piece, and when you have got hundreds of videos, let's just say I sat there clicking buttons and watching like 3 seasons of New Girl after the field season was over. So we look at these finer scale approach behaviors because in 2 different trials, a female may have a very similar path in getting to her choice, but she may have very different ways of covering that path. So the female may take very, very long hops, very straight movements towards her choice, or she may really take small hops, crawl a little bit, take her time, and really make sure she wants that call. So, now that we've got kind of background into what we're doing with this, our second chapter looked at how natural variation in light levels affected her preferences. So, I had 2 sets of treatments here. I had a new moon treatment, which is about what starlight is in 0.2 lux, and then a full moon treatment at 2 lux. So we are looking at those 2 different phases of the moon, and our hypothesis was that changes in nocturnal light levels associated with the changing phases of the moon affect female tree frog behavior in our study. So our prediction was that under bright moonlight, females would show behaviors that mitigate the increased risk of being detected by predators, such as relaxing their preferences, decreasing their choosiness, as well as showing more directional, faster, or stealthier movements towards their choice. So, we found that variation in moonlight does not change her preferences. So you can see here that we've got our two different phases. Our black line represents our new moon, and our gray line represents our full moon treatment. And we've got that preference function all the way from our unattractive 6-pulse call, our Logan call, to our 30 pulse, which is KJ Appa, I think is how you pronounce his name. Again, I barely know these people, but we look at her response across that, and you can see that although there's a variation in the shape of these preference functions, she is still preferring that attractive male call over her unattractive option. So our stimulus itself, the call duration, is significantly different. However, our treatment in Lux did not make an impact, and you can see that atrocious feedback there. So, going back to our preference traits, we also found that these are very startlingly similar. So her highest response to those traits was the same across those light treatments, again with her strength, her tolerance, and her overall level of responsiveness were not significantly different across those two light treatments. We also found that her choosiness did not change in this normal light range. So you can see again, in our black line, which is our new moon treatment, and our gray line, which is our full moon treatment, the number of females choosing, or the percentage of females choosing the attractive call is always going to be 100% at the beginning when they are equal distance apart. And you can see that right around between 9 and 12 decibels, so you're pulling him further and further away, you see that switch where she goes, he's just not worth it. And she's stopping and going towards that more unattractive call more. So you can see again that there is that very distinct shift. So our attenuation is significantly different. However, our treatment did not make an impact on how she did that. So she didn't stop at some point in a different way across those two treatments. We also found that the variation in moonlight does not change her movement behavior. So her number of movements, her head turns, crawling, and hopping behavior was not significantly different in our new moon and full moon treatment, as well as the time she took to make her first move, as well as the time between moves, and her distance in centimeters per move. So these are all pretty similar across our two light conditions in the natural nocturnal range. So, some conclusions from this. Well, how does lunar-mediated behavior affect sexual selection? In short, it really doesn't in our species. So therefore, it does not affect the strength or direction of sexual selection. Now, there is a point I'd like to mention here, that other species have shown preference shifts with variation in night levels. The two very prominent studies on this is in tungura frogs and reed frogs, and these authors found that females were moving more in the brighter conditions and were moving from approach perches more in those bright light conditions. And both of these authors attributed that to females feeling safer in the dark. Oh, I think I messed that up. Just kidding. Opposite. They were moving more in the dark. So they were feeling safer in those dark conditions. So what about our species? Well, there was a comparative study done in around 2012 that looked at a vast variety of species, and what they did was they took research studies across the board and found that some species showed very strong lunar effects. Others didn't really respond at all, and then the ones that did show lunar effects had completely different responses. So, why are our females different? Well, to start, we have a very short breeding season. The studies on tungura and reed frogs are done in a tropical species. So these species can breed for a very long time, whereas our poor little things in Wisconsin are subjected to our very cold Wisconsin spring and early summer. So they're having a longer time to get into the warm weather where they're actually having favorable breeding. So they're subjected to a lot stronger environmental factors, such as our temperature and rainfall. So this was a graph that was actually created by one of my undergraduates. I had a tendency in grad school to pluck my really good students from my teaching sections and bring them into my lab to do some great research. This one was done by a grad student I had named Rachel, and she did some wonderful studies where she looked at our average temperature, which is plotted in red, and our rainfall, which is plotted in blue. So you can see that when we have our high choruses nights are when you're seeing these temperature spikes at the beginning of the season. However, once you reach around July, you're getting those hotter temperatures, and our females and males aren't really coming out to breed anymore. It gets too warm for them, and then you have a cutoff, and these temperatures are getting too high. So our frogs are affected by both having that shorter breeding season of about 8 weeks, which is getting even shorter with our change in our climate. So last year, my advisor is actually having to start considering doing an entirely different species because our species has gone from a prolonged breeder over 8 weeks to an explosive breeder, where they finally get that warm weather and they are coming out in full force, but not really coming out the rest of the season. So my first season of collection, we had probably 20 collection nights across May all the way to July. My last field season, we had 5 collection nights. And those were basically all the first week or two of field season. Let that sink in for a second. That's a huge shift in breeding behavior. So again, any more on this, you can see in my second paper. And then we jump into the third, looking at ecological light pollution affecting female preferences. So we found no significant differences in their behavior across the spectrum of our natural moon phases. So what about light pollution? Our natural spectrum is again about 0.2 lux to 2 lux. So how would female preferences change under increased light levels? One pollutant that has been found in many different studies to be of particular importance is artificial light, especially to our nocturnal animals, and you can see why. So this picture is even from a couple of years ago, and you can see that as we as humans are urbanizing areas, we are creating a lot of light. And ecological light pollution includes chronic or periodically increased light levels. So chronic, you have a continuous stream such as stadium lighting, porch lighting, security lighting, basically any lighting that's going to be consistently on, and periodically increased light levels, a source that is going in and out such as car headlights. And light pollution has been shown to affect the behaviors of many different species and has been shown to affect their foraging, reproduction, communication, and locomotion. Let that sit in for a second. That's like everything they could possibly do. So them eating, them reproducing, them speaking to each other and getting valuable information, as well as them even moving around. So basically anything an organism can do can be affected by this artificial light. That is a huge conservation problem. So my third chapter looks at how ecological light pollution affects female preferences. And again, we have 2 different experiments. I separated this into 2 different experiments with 2 completely different treatments. The first looked at our static light conditions, so our constant continuous light source. And I wanted to include in here something at the natural level, so I used our new moon treatment light level of 0.2 lux, a static light pollution level of 5 lux, and then a light pollution level of about 15 lux, which is about the lighting in this room right now. My experiment 2 set up static versus dynamic light pollution. So we had a treatment of 15 lux, our maximum light treatment there, and then a dynamic light pollution which mimics the movement of car headlights. So a movement up to 15 lux and then a quick off as if the car is passing you. So our hypothesis for this experiment overall was that anthropogenic light pollution affects mate choice behavior of female eastern gray tree frogs. And we had 2 different experiments, so 2 different sets of predictions with one very key difference. The first looks at how higher light levels and how that could result in relaxed preferences, decreased choosiness, and that more directional, faster, or stealthier approach movement towards her choice. And then experiment 2 looked at how fluctuations in the light levels would result in those same predictions. So decreasing their choosiness, relaxing their preferences, and that more directional, faster, or stealthier approach movement towards her choice. So in our first experiment, chapter, so you can see that our 3 light treatments are low, medium, and high, or 0.2, 5, and 15 lux. We're set on that call duration stimuli, just like our first set of experiments, and we can see that females are preferring that 30 pulse, more attractive male call over their 6 pulse, less attractive male call in all 3 of those light conditions. And again, although those are very different shapes of their preference functions, it's not significantly different. So our stimulus, our call duration, is still significant. She's showing that preference for the longer calls over the shorter calls. However, she does not have a difference in those different light treatments. And the same is found with our preference functions. So you can see that she is performing, having that peak preference about the same across our 3 light conditions, as well as her strength, tolerance, and overall response level. We also found that the static light pollution does not change her choosiness. So again, you're seeing that the females are choosing that attractive call 100% of the time. And again, right around, this is a little bit further, so we pushed it to around 12 or 15, that just died. 12 or 15 decibels, we're seeing that shift where she's going to move towards that unattractive male more. So that attenuation is significantly different. I completely died here. So you can see that she is moving towards that unattractive male, so our attenuation is significantly different. However, it is not different across our light treatments. And we found the same in our dynamic light pollution as compared to our static. So in our dynamic and static, again, very different preference function shape here. You can see that our dynamic has that beautiful preference function curve, whereas our static has a pretty straight and linear line there across the call duration of 6 to 30 pulses. And again, our call duration is significant. She is showing a preference for that 30 pulse call over our 6 pulse call. However, our light treatment did not make a difference there. And again, our preference traits are nearly identical in this one for her peak strength, tolerance, and her overall responsiveness to that call. And the dynamic pollution was also found to not change her choosiness. So again, you've got that shift in her attenuation between 9 and 12 decibels, where she is choosing that attractive call, and then around 9 to 12, she says he's not worth it anymore. And again, a slightly different shape in our curve, however, it is not significantly different. So the last part, and I like to include a little animation here to show you kind of what our different light treatments look like. So we've got our constant static source that is constantly on, as compared to our dynamic, which moves in and out, which was crafted by my undergraduate with a light dimmer. And we found that her latency to leave, her choice latency, and the number of crawls that she made were not statistically different. So you can see in here, this graph is a little bit hard to read. You're following the treatment across, so our thin line is our 6 pulse call, and our thick line is our 30 pulse. And you're gonna look at it from left to right for our static to dynamic. And you can see that her latency to leave in both the 6 pulse and the 30 stays somewhat in that range, as well as her choice latency and the number of crawls. However, she had a very different number of hops on the 6 pulse and the 30 pulse call, where she dropped the number of hops she was making in that dynamic treatment very drastically. I'm not quite sure why that happened there. So the very last part of our movement analysis that we did for this study is we wanted to make sure that the females were not moving more or less than we expected that we actually observed in the study. So this is somewhat what our pattern of our light went. So we were majority dark. So when a car is approaching you, the light is slowly increasing, and then as soon as it passes you, it cuts out right away. So our trials spent approximately 70% of the time in the dark and about 30% of the time in the light. So we wanted to ensure that the females were not moving more in the dark than we expected in both the 6 pulse and the 30 pulse call. And we found that they were staying pretty consistent. That's pretty close as far as expected versus observed there. And that was just a check to make sure that our own experiment wasn't causing bias. So from this, we concluded that light pollution associated with hinder female mate choice in our species. So, are we able to say based on this experiment that light pollution is unimportant for amphibian conservation? Not quite. The effect of light on mate choice behavior may be highly species specific. As I mentioned before, that researcher put a mass study out there that looked at all of those different species of frogs, and she found that some have effects, some have completely different effects across the phases of the moon, with some being more active in the light, whereas some are more active in the dark. And then some species, like ours, don't really care at all and have no effects by the moon phase. So, in order to push that, we can say maybe if she's more active during our natural phases, she might be more active in our light pollution conditions, or the opposite. So, a knowledge of a species' response to variation in our natural light range may actually help us gauge how vulnerable they are to anthropogenic light pollution conditions. And again, any more you would like to read on this can be found in my third paper. So before I end, I do want to have some future research suggestions. I think this is a very powerful source in science that you can do. In a neuron mate choice experiments, so frogs and toads, all trials are conducted in a comparatively small playback arena. So you guys saw in my video, that's our whole experiment. It's done in a very short range, and when you think about the amount of distance that a female is going to cover to even get to that pond to mate, there's a lot that we don't see when we conduct that in a very small playback arena. So ideally, we would like to see more of her behavior, because our observations currently only encompass the last few meters that she's going to approach that male. Unfortunately, this would also require us as field scientists to get out there and try to follow a female frog all the way from the woods to the pond, and I will tell you, those females dive under the water and you will never find them again. So that is a huge challenge in why we conduct these experiments the way we do right now. But as technology increases, radio telemetry data, we could do a lot more with this as we get more technology. And because of all this leaves a lot of factors of female mate choice behavior unexamined, but again, as we get more technology, we can increase this even more. So with that, I'd like to throw some acknowledgements out there. First, I'd like to thank my crazy advisor, Gerlinde Höbel. There were a lot of very fun times in the lab, and a lot of times where I was super stressed out and made to go out and collect frogs in a lightning storm. I'm not kidding. I'd also like to thank my committee members, Dr. Rafael Rodriguez-Sevilla and Dr. Peter Dunn, as well as my lab mates that went out with me every night to collect frogs, even though we were over-caffeinated and very exhausted. Another shout out I'd like to make at this point is the designer of our preference function program. All those beautiful preference functions were from a program called PFUNC that was created by a graduate student named Joey Kilmer. If you are interested in the biology and computer science crossover, he is amazing. Like, he went and started a biology PhD and ended up writing papers and writing a program to help make our lives as scientists easier as far as analysis. So he's done some really great things with the biology computer science crossover. If that's something that interests you, I highly recommend checking out his work. And then, of course, the plethora of undergrads that have been throughout there. Rachel Greminger was the one that did that study on light levels, or not light levels, precipitation and temperature and how that affected core size, as well as Lindsay Hayward, which sat there, helped me with the dimmer as we sat from 9 p.m. to 5 in the morning testing frogs. Yeah, I'm nocturnal like my species sometimes. So all of those students and my committee members helped me become the scientist I am today,